Section: Introduction. In this section, we discuss how critical advancements in science and engineering rely on our ability to analyze data across different scales. Instead of focusing on individual data points, we need to consider the entire populations from which these points are drawn. For instance, in areas like single-cell genomics and DNA design, the key analysis unit is the distribution of samples, such as the state of a cell or the patient it comes from. These scenarios are hierarchical. We observe groups of samples from underlying distributions, which are in turn derived from a broader meta-distribution. If we don't model these distributions directly, we risk losing important population-level signals amidst the noise. Our main challenge is to learn representations that capture these distributions rather than just individual data points. We introduce generative distribution embeddings, or GDEs, which enhance autoencoders to work within the distribution space. In our GDE framework, the encoder transforms a set of samples, an empirical distribution, into a latent space, while the decoder is replaced by a conditional generative model that reconstructs the distribution by sampling from this latent representation. This approach naturally accommodates hierarchical data modeling, treating each observed set as a sample from a latent distribution. A key insight is that we can learn strong distributional representations by combining conditional generative models with encoders that meet a minimal distributional invariance requirement. We support this idea through a statistical decision theoretic perspective. Our framework integrates concepts from conditional generation, empirical process theory, and information geometry. Empirically, we demonstrate that GDEs function as approximate predictive sufficient statistics, effectively capturing distribution-level structures while filtering out sampling noise. Additionally, the latent spaces we learn show geometric regularity. Distances in this space correlate with Wasserstein distances between distributions, and linear interpolation in the latent space resembles optimal transport paths for distributions supported by the meta-distribution, especially for Gaussian types. We validate GDEs using synthetic datasets with known structures, showing that they outperform baseline methods in terms of generative fidelity and structural preservation. We then apply our approach across various domains in computational biology, highlighting GDE's flexibility in modeling distributions defined by different organizing principles, such as distinct populations and varying experimental conditions. We present 6 applications: learning representations of cell populations from lineage tracing data involving 150,000 cells, predicting single-cell transcriptomic responses to changes with 1 million cells, predicting cellular phenotypic responses with 20 million cell images, modeling tissue-specific DNA methylation with 253 million sequences, designing synthetic promoters with 34 million sequences, and modeling spatiotemporal distributions of viral protein sequences with 1 million sequences. Across these applications, GDEs provide a robust and scalable framework for making inferences at the distribution level. We also make our code available for all experiments. In our methods, we observe multiple sets of samples, where each set is independently drawn from an unknown distribution. Our goal is to learn an encoder-generator pair that can encode a finite sample and decode it to yield a distribution close to the original. This requires the encoder to extract structural information from noisy observations, leading us to impose 2 key constraints: permutation invariance and proportional invariance. Together, these ensure that the encoder relies solely on the empirical measure of the samples. In addition to the encoder, we need a generator that can learn to regenerate the distribution. We can adapt any conditional generative model to minimize the divergence between the true distribution and the generated sample. This unifies our approach to conditional generation and representation learning for distributions. We provide a theoretical justification for this objective in the appendix. Section Summary. In this section, we introduce generative distribution embeddings, GDEs, a framework that enhances autoencoders to model distributions rather than individual data points, enabling us to capture hierarchical data structures effectively. We demonstrate that GDEs can learn strong distributional representations through a combination of conditional generative models and encoders, leading to improved generative fidelity and structure preservation across various applications in computational biology. Section: From labels to distributions. In this section, we discuss how we transition from labels to distributions. In many real-world scenarios, we work with a dataset consisting of pairs of data points and their corresponding labels, rather than directly from a distribution. To utilize our framework, we organize data points into groups that represent empirical distributions, which approximate draws from a target distribution. This grouping is informed by our understanding of the label space structure, allowing us to shape the latent space for further applications. When the label space is discrete, we can create groups by clustering data points with the same label, such as digits in the MNIST dataset or cells categorized by their identity. If there are similarities among discrete labels, we can form groups based on those similarities, similar to how contrastive learning defines neighborhoods based on labels. For continuous or structured labels, like spatial coordinates or temporal sequences, we can use a similarity measure to select points close to a target label, creating a probabilistic neighborhood that respects local structures. In cases where labels are noisy, we can adjust for this by sampling a latent target and calculating weights that reflect the uncertainty in the data. All these methods can be unified under a common framework where we draw from a prior distribution over labels, compute weights based on the empirical distribution, and sample data points accordingly. This framework encompasses the examples we discussed and provides us with tools to shape the latent space effectively. We also review related work in the field. Various studies have explored learning distribution embeddings or summary statistics. Kernel methods, like kernel mean embedding, offer non-parametric ways to represent probability measures, which can be used for tasks such as regression and classification. Our approach naturally incorporates these methods as specific cases of our distribution ally invariant encoders. Additionally, we note that some works model distributions geometrically, treating them as points on a manifold with a specific metric. However, many of these methods are not generative or are limited to categorical distributions. Other research has focused on learning flows over Wasserstein spaces, primarily for transport problems, while our goal is to auto-encode distributions. We highlight that our framework, GDEs, complements existing methods and can be integrated into various approaches. One recent method, Wasserstein wormhole, represents distributions in a way that aligns Euclidean distances with divergences in the sample space, serving as a specific instance of GDEs. We also differentiate our work from methods that learn summary statistics in supervised settings, emphasizing that our framework aims to resample encoded distributions. We provide theoretical guidance for designing GDEs and demonstrate that distribution embedding is closely linked to generative modeling, allowing us to create high-quality GDEs for complex problems. On the architectural side, our encoder requires a model that is invariant to distribution changes. While we introduce the concept of distributional invariance, it builds on the well-studied idea of permutation invariance. Some existing methods are permutation invariant but not distribution alley invariant, while others, like mean pooled attention layers, meet both criteria. A significant contribution of our work is the realization that any conditional generative model can be adapted to learn distributional representations. Recent findings in the vision domain show that conditional diffusion models can produce strong representations. We formalize and expand on this idea, demonstrating that various modern techniques, including variational autoencoders and denoising diffusion models, can be utilized to learn GDEs. Our framework is flexible enough to incorporate any future conditional generative modeling approaches as well. Section Summary. In this section, we describe how to transform datasets into sets that approximate underlying distributions, allowing us to shape the GDE latent space based on label structures, whether discrete or continuous. We also discuss how our framework unifies various methods for learning distribution embeddings and highlights the flexibility of GDEs in integrating with existing generative modeling techniques. Section: Statistical and Geometric Properties of GDEs. In this section, we explore the statistical and geometric properties of generative distributional embeddings, GDEs. A key aspect of GDEs is their ability to separate observational noise from the true structure of the underlying distribution. We approach this from two angles. First, by learning predictive sufficient statistics, and second, by embedding statistical manifolds. Our main goal with GDEs is to accurately recover the true data generating distribution from limited, noisy samples. This involves creating a representation that distinguishes the actual signal from the noise. We define this goal through the concept of asymptotic predictive sufficiency, which means that as we gather more samples, our statistic should closely approximate the true distribution for new observations. In our case, the encoder we use acts as this statistic, effectively determining the underlying distribution. We demonstrate this with the multinomial distribution, where we learn GDEs for three-dimensional multinomial distributions using a graph neural network encoder and a conditional diffusion generator. The latent space we create captures the structure of the multinomial simplex. We also apply this to real-world datasets with discrete class labels, such as a subset of MNIST and synthetic DNA sequences. In both cases, we find that GDEs recover the same underlying structure in the latent space, despite using different domains and architectures. The geometry learned across these experiments is remarkably similar, with distances in the latent space closely resembling those calculated using a Gaussian approximation. Next, we present a geometric view of the GDE problem through optimal transport. We define our sample space and consider the set of probability measures with finite second moments. The 2-Wasserstein distance helps us understand the minimal cost of moving mass between distributions, giving us a geodesic structure. We can interpret paths between distributions as geodesics in a manifold, which allows us to define a statistical manifold based on our meta-distribution. We can think of our encoder as a smooth embedding of this manifold, preserving its local structure. While we don't expect GDEs to maintain the Wasserstein metric perfectly, our empirical findings suggest they behave like approximate isometric embeddings. For instance, when we train GDEs on samples from two-dimensional Gaussian distributions, we observe a strong correlation between distances in the latent space and true Wasserstein distances. Similarly, with Gaussian mixtures, we find a high correlation as well. These results indicate that GDEs effectively summarize noisy data and maintain the geometric properties of the statistical manifold. Section Summary. In this section, we explore how generative distributional embeddings, GDEs, effectively separate observational noise from the underlying data-generating distribution by learning predictive sufficient statistics and embedding statistical manifolds. We demonstrate that GDEs can recover consistent representations across diverse domains and maintain a geometric structure that correlates well with optimal transport distances, indicating their ability to summarize noisy data and preserve the geometry of the statistical manifold. Section: Learning task-specific geometries by prior weighting. In this section, we explore how to learn geometries specific to tasks by using prior weighting. Traditionally, sufficient statistics and statistical manifolds are seen as fixed, determined by the distribution family rather than learned from data. However, we learn an embedding from samples taken from distributions that come from a broader meta-distribution, which we denote as Q. This means that the representations we create, along with the geometry they form, are influenced by Q. In previous examples, we used a roughly uniform Q over a part of the statistical manifold, M, and found that the learned geometry closely resembles that of Wasserstein space. Now, we show that when Q is non-uniform, it distorts this geometry, allowing us to create task-relevant structures in the latent space. Specifically, areas with higher prior density expand to maintain finer distinctions, while areas with lower density contract. We confirm this in a synthetic multinomial context by training GDE models on empirical distributions sampled from skewed Dirichlet priors, comparing them to a uniform prior. As anticipated, distances stretch in areas where Q is concentrated. This indicates that the geometry we learn is influenced by the prior, reflecting both the intrinsic distances of the distributions and the sampling density from Q. Prior weighting is not just a minor detail, it is a crucial tool. By strategically selecting Q, using task-informed sampling strategies, we can shape the latent space to highlight meaningful distinctions for our objectives. Next, we benchmark our approach and demonstrate the versatility of GDEs across various biological science tasks, including DNA sequences, protein sequences, gene expression data, and microscopy data. We investigate different combinations of encoder-generator pairs, with a detailed discussion of architectures and training dynamics provided in the appendix. To benchmark GDEs on synthetic distribution datasets, we note that the design space for GDEs is extensive, allowing any distribution ally invariant encoder to be paired with any conditional generative model. To inform our implementation choices, we systematically evaluate architectures using synthetic datasets. Our benchmarks include two existing methods that GDEs generalize: kernel mean embeddings and Wasserstein wormhole. We assess 30 combinations of encoders and generators on multivariate normal distributions in 5 dimensions. For evaluation, we calculate the Wasserstein reconstruction error by estimating means and covariance matrices from generated samples and applying the closed form for the Wasserstein distance between Gaussians. We find that mean pooled graph neural networks with skip connections paired with DDPM generators yield the highest quality generations, surpassing existing methods. We present results for this architecture on synthetic distributions, with full experimental results available in the appendix. Additionally, we benchmark this GDE architecture on 3 more complex datasets. 1. 3-component Gaussian mixtures in 5 dimensions. 2. Mixtures of MNIST images based on categorical distributions of 3 classes. And 3. A similar dataset using fashion MNIST. For image datasets, where calculating the Wasserstein distance is not feasible, we compute the synchrone divergences between pre-trained ResNet 18 representations of generated and true samples. In all scenarios, our selected GDE architecture outperforms existing techniques, including kernel mean embeddings with DDPM for generation and Wasserstein wormhole. Finally, we address modeling clonal populations in lineage-traced single-cell RNA sequencing, screen-seq data. While many methods exist for learning representations of individual cells from screen-seq data, approaches for learning representations of cell populations are less developed. This is particularly relevant for analyzing lineage tracing data, where we focus on clones, or groups of cells from the same progenitor. Using lineage-traced screen-seq data from mouse hematopoietic stem cells, we apply GDEs to learn representations at the clone level by treating the cells within a clone as samples from an empirical distribution. Following established frameworks, we assess how well these representations can predict future clonal gene expression by examining the mutual information between a clone's representation at an early differentiation stage and its representation later on. We find that GDEs with a conditional variational autoencoder generator outperform Wasserstein wormhole embeddings by over 2 bits. We then investigate whether this improvement in predictive power is due to enhanced representations within specific cell types, such as neutrophils or monocytes. By breaking down mutual information estimates into their individual contributions, we discover that the contributions span the entire cell state space rather than being limited to any specific cell subtype. Section Summary. In this section, we learn task-specific geometries by prior weighting, demonstrating that non-uniform priors can warp the learned embedding geometry to reflect meaningful distinctions in latent space, which is crucial for downstream objectives. We benchmark our generative distribution embeddings, GDEs, across various biological data tasks, showing that our architecture outperforms existing methods in generating high-quality representations, particularly in lineage-traced screen-seq data. Section: Predicting transcriptional responses to genetic perturbations. In this section, we focus on predicting how genetic changes affect gene expression. We assess the performance of GDE in predicting these effects using perturb-seq data, which captures gene expression responses to CRISPR knockdowns of many genes. We set up a task to predict the full range of transcriptional responses based on the type of genetic change. We compare 2 methods, one where we train a linear model to directly predict the average expression profile, and another where we predict the GDE embedding of the expression distribution and then derive the average from this embedding using a learned linear projection. In both methods, we apply ridge regression on gene PT embeddings to allow for generalization across different genetic changes, showing that GDE enhances both the R-squared and mean-squared error metrics. Next, we apply GDEs to analyze pooled image-based CRISPR screening data, which examines the effects of altering 5072 essential human genes in HeLa cells. This dataset contains over 20 million single-cell images, revealing a wide range of phenotypic variations. Each genetic change leads to a variety of cell shapes, which we treat as empirical distributions and use to train a GDE model. We explore different inductive biases by using two types of priors, one that models the spatial structure of images and another that captures variations across genetic changes. Our model successfully learns to replicate key phenotypic features, such as nuclear shape and cytoplasmic texture. We validate our approach by holding back 30% of the most impactful genetic changes and using ridge regression to predict the GDE embedding, achieving strong generalization with an R-squared of about 0.71 and a low mean-squared error. We also demonstrate how GDEs can analyze bisulfite sequencing data without the need for extensive preprocessing like genome alignment. We show that GDEs can identify tissue-specific DNA methylation patterns directly from sequencing reads. By simulating RID distributions based on known methylation data, we allow the GDE model to learn without providing explicit methylation information or a reference genome. We use a 1D convolutional network for encoding and a hyena DNA model for decoding. Our method processes large volumes of reads efficiently, enabling the model to learn methylation signatures from specific tissue distributions. We achieve a test accuracy of 60% for coarse tissue classification and 35% for fine-grained classification. Lastly, we evaluate GDEs on a large dataset from a reporter assay that measures transcriptional activity across 34 million yeast promoter sequences. Each promoter consists of a random DNA sequence tested for expression in yeast cells. Since the sequences are randomly generated, we cannot rely on shared structures, but rather on how variations in sequences affect expression levels due to transcription factor binding sites. We create a learning task where each example consists of sequences from a specific expression range, holding out the top 5 ranges. We train a GDE using a 1D convolutional network for encoding and hyena DNA for decoding. The GDE embeddings we learn show a smooth transition across expression levels, and we can identify the transcription factor motifs present in both real and generated sequences, indicating that our model captures biologically relevant variations. Further details can be found in the appendix. Section Summary. In this section, we evaluate the GDE model for predicting transcriptional responses to genetic perturbations using perturb-seq data and demonstrate its effectiveness in reconstructing morphological responses from CRISPR screening data. We also show that GDEs can directly analyze bisulfite sequencing reads to identify tissue-specific methylation patterns and decode yeast promoter sequence activity, achieving strong performance in both tasks. Section: Modeling spatiotemporal distributions of viral lineages. In this section, we present our modeling of the spatiotemporal distributions of viral lineages. We have developed powerful approaches to represent individual protein sequences, and we demonstrate that the GDE framework can effectively extend these methods to learn about distributions of sequences. Specifically, we focus on the distributions of SARS-CoV-2 spike protein sequences over time and across different locations. Using data from the Global Initiative on Sharing All Influenza Data, GISAID, we categorize sequences by the month they were sampled and their site location, treating each category as an empirical distribution of protein sequences. We utilize a GDE that combines the ESM architecture with a mean pooled graph neural network, GNN, as the encoder and a conditional ProGen2 architecture as the generator. Our results indicate that the learned latent space organizes samples in chronological order, suggesting that GDEs effectively capture the time-varying nature of sequence distributions. Quantitatively, we find that ridge regression on GDE representations predicts the month of unseen sequence distributions with a mean absolute error of about 1.83 months, which is an improvement over the baseline mean pooled ESM embeddings that have a mean absolute error of about 2.24 months. We also observe a weaker spatial signal, where a support vector machine, SVM, trained to classify distributions by country achieves an accuracy of about 0.28 from GDE representations, compared to 0.25 from mean pooled ESM embeddings. Both methods slightly outperform the baseline accuracy of 0.21, which predicts the most common label, USA. In our discussion, we introduced generative distribution embeddings, a framework that combines distribution invariant encoders with conditional generators to learn structured representations of distributions. We show that finite sample sets can be mapped by smooth embeddings that identify the underlying distribution, allowing for consistent reconstruction as sample sizes increase. We formalize these properties through connections to predictive sufficiency and statistical manifold embeddings, proving that a wide range of encoder architectures can be asymptotically normal and unbiased when trained with a plug-in loss. We have demonstrated the versatility of GDEs across various large-scale biological problems, showcasing their ability to work directly with measurement data while modeling population-level structures. Importantly, GDEs allow for flexible distributional constructions, indicating that many problems can be framed as population-level modeling tasks. We provide code for model training and dataset preprocessing. However, we acknowledge some limitations. GDEs depend on careful choices of meta-distributional priors, which often require domain-specific design. They also present practical engineering challenges, such as propagating gradients through the generator and scaling to large set sizes. The current theoretical framework assumes exchangeable samples and does not accommodate non-independent and identically distributed samples within a distribution. While we provide empirical evidence that GDEs learn isometries across domains, we do not yet have mechanistic proof.